Alright. So, let me just make sure everything's good. Oh. Okay. Hopefully this works. If you can't hear me, I'm sorry. Alright, so exam 4, the written portion. We got, what we got up here? We got, there's a question mark. Okay. So below are some lac operon-like genotypes of certain strains of E. coli. Indicate whether they are inducible, non-inducible, or constitutive. Assume that the glucose levels are low in all cases. For partial diploid strains, D and E, describe the, excuse me. Describe the overall phenotype of the cell. Don't provide a separate answer for each copy. Note that IS is dominant to I plus is dominant to I minus, or is it L? I can't tell. Probably L for lac. And OC is dominant to O positive. So, we got our first one up here. It's, let me see if I can highlight. You got L negative, P positive, O positive, Z positive, Y positive, and A positive. So Z, Y, and A are structural for the lac operon. And in almost all cases here are positive. So really we're going to focus, at least for the first 4, on the I, P, O, and Z. Or I, P, and O, or L. So L will be the lac gene itself. The P is the promoter, and O is the operator. So in the first one, I or L is negative, P is positive, O is positive. Well, the I is negative or L is negative, whatever. We're just going to call it I for the rest of the video. If I is negative, then the whole thing's shot anyways, because normally it needs to be positive, and then there to be an introduction of lactose for it then to be inducible. However, because it's negative, it is non-inducible because it doesn't even operate if there is any lactose in the environment. So IS, and then the rest positive. This means that the repressor, after a single cycle, will bond to the operon or operator portion of the operon and repress the further operation of the operon. So it won't work. So it's non-inducible as well. C. In this one, the promoter is negative. There's a defect in the promoter. So even if there is lactose, because the L is positive, because the promoter is negative, the ship won't sail. It won't make anything. Even though the O, the operon, is unable to have a repressor bind to it because the promoter doesn't work in the first place, it never starts. D, we have IS for our plasmid and OC for our chromosome. So normally, like earlier in B, there's IS, so the repressor will bind to the operon and never come off, which would make it non-inducible. However, in the chromosome, the operator, the OC, the LAC-O, it has that C next to it, which means it cannot have a repressor bound to it. So it's constitutive. It was just constantly running. It can't shut off because the repressor can't bind to it. So, yeah, that's the phenotype is constitutive. For E, this is an exception. In the plasmid, we have a structural anomaly here, Z, which makes it beta or S-sect galactosidase, which helps to make allolactase toast, is messed up and is deficient. And the chromosome is I negative, so it won't initiate at all. So we can't even look at the chromosome because even if something did work, it won't make anything. in a normal circumstance, but it will in this case be inducible, because even though the plasmid cannot get the repressor off of it, so it'll only go I and P, and then the O is messed up, the plasmid can still transfer its lactose to the chromosome and have it work. So it's inducible, it's inducible, not constitutive, because there needs to be lactose for this to work. Okay, anyone confused yet? Moving on to 2. 2. We want to use the Sanger method to do DNA sequencing of this following string of code. And we have 5', the mRNA, the primary annealing site, and then the 3'. So the primary annealing site indicates the region in which the template DNA strand, where the primer will anneal. And so during the sequencing reaction, in order to generate newly synthesized strand, and so note this newly synthesized strand will be complementary to the sequence above. So complementary, it will be 3' dash that sequence except all the A's will be T's, T's, A's, C's, G's, G's, C's in this reaction. And so using the electrophoresis on this gel, or mark out on a table what's going to happen. So what you do is you make 4 columns. At the top of each column you have DDGTP, and then DDATP, then DDTTP, and then DDCTP. Okay? And then you write the complement going top down of that sequence. So it would be TAGCCACTGCAATACG. So, and then 5', or 3', 5'. And then for the corresponding row, if the first one's T, then we go over to our 4th column, DDTP, and mark it. And then just go down the list. So for A, it'll be GA, mark it. And then the next one's G, so it'd be the first row, mark it. And so you get this kind of like scattered X's throughout, but really it just is the same way as coding, writing the code for the supplement on this one. Here, maybe I can switch to this for a second. Awesome. Okay. So you get something that to the right. Everything's opposite here. You get something like that, Oh, then fix that. So there's our code, and then along that column there, you see how it's inverted, and then you get the X's just or dashes right as those genes come up. So that's how that works. Alright, 3. 2 mRNA codons below A and B are referenced. Consider the wobble rules and give all possible tRNA anticodons that combine to the following codons. Be sure to indicate the polarity of each anticodon. So for A, there are 2 possibilities, which would be 3' CGG 5', and 3' CGI 5'. So there's only 2, because if you look up at our modified rules here, it says, when it ends in your C there, it can only be G. And then, because the third one's C, we can also have an I. So those are the 2 possibilities. Even though, as a vanilla rule, there's 5 possibles, because the first 2 letters stay the same, and the last codon, or the last nucleotide, can switch around. There's additional rules that go with it that cancel the other 2 out. So it can't be CGA or CGU by those rules. So for B, AAG, again, 3' will be UUC or UUU. Again, going 3' to 5'. And those follow the same rules. It ends in a G here. And so I'll end up with a C and all U's. Okay, so the two TNR anticodons below UAU and AUG, what anticodons combine to it? So this one kind of go backwards, right? You got your tRNA that ends in a U, and so you got AUA is what goes with it. 5' to 3' AUA. Again, according to this table of rules. And then, what else we got here? We got AUG. And AUG will be 5' CAU. Again, according to the table of rules here. Now, for an average mass, now this is a fun question. I had fun doing it. I don't know if I got it right, but I had fun with it. The average mass of 20 common amino acids is 125 Daltons each. So that's not saying 20 common amino acids is 105 Daltons. It's saying per amino acid is 125 Daltons. So 20 of those is 2,500 or 25 kilodaltons. Fun stuff. Suppose E. coli protein called YFP is 75 kilodaltons. So what is the length of nucleotides in the portion of the E. coli mRNA that is translated to make YFP? Well, if it's all mRNA, then you take the 75 kilodaltons divided by the 25 kilodaltons, and you get 3. So there's 3 sets of 20, or 60 amino acids, and these acids are comprised of 3 nucleotides each, so there would be 180 nucleotides. So 180 should be the length of nucleotides, the portion of the E. coli mRNA that is translated to make YFP. But of course, that's not including untranslated regions, which are a range. So I'm not sure how to calculate that. But so B, the E. coli that makes YFP contains 5 UTR and 3'. What's their purposes? They are the untranslated regions which act to regulate transcription. They, depending on the organism, in this case E. coli, operate to ensure that not everything gets copied at the same time, but only what's needed for the different functions. So that's called UTR, untranslated regions. And there's one immediately before, or preceding and following UTR, or decoded mRNA. So how many ATP molecules are required to translate 100 copies? Well, it's 1 ATP per 1 ATP per amino acid. So that's 60 times 100 copies, or 6,000 ATP. And for D, for 50 copies, how many is GTP? Well, you have 2 for initiation, 2 for elongation, and 1 for termination. So for 1 copy of YFP, you would have 120 GTP. 120 by 50 is also 6,000. So 6,000 GTP. Same answers for C and D, except, -1 triphosphate. Anyways, so hope that was understood. I probably slur really badly. I got this canker sore on my tongue, so you don't want to know that, but speaking a little off. Alright, see you around.